Hello guys and welcome back to Excel Academy. So as we were discussing about the regulation of glycolysis, so today the third step of the glycolysis, the regulation we are going to discuss, that is the last step in the glycolysis, that is your pyruvate kinase. So if you remember from the previous lecture, so we have got 10 steps in the glycolysis, right? So among these 10 steps, there are 3 which are irreversible. Okay. So first one is with the hexokinase. So you have got the regulation at the hexokinase level. Second, you have got at the phosphofructokinase where fructose 6-phosphate is converted to fructose 1,6-phosphate. And the third one is with the pyruvate kinase. Okay. So already we have discussed about the rest two, and today we are going to discuss the regulation of glycolysis based on the pyruvate kinase. Okay. So pyruvate kinase is the third regulated enzyme of glycolysis. It is regulated by various allosteric effectors like AMP, ATP, acetyl CoA. So PK, we have got AMP, ATP, and acetyl CoA. So Jani, tell me what will happen to PK, whether it will increase or decrease if I have got AMP more in my cytoplasm.
It decreases, ma'am.
The PK activity?
Decreases.
Sure.
Yes, ma'am.
Why it will decrease?
Because it converts ATP, no, ma'am.
No, it
AMP
does not
to
convert.
ATP.
It does not convert. What does pyruvate kinase does? It is going to convert your phosphoenol pyruvate to pyruvate. It converts your phosphoenol pyruvate to pyruvate, plus it generates ATP. So what will happen if there is a increased concentration of AMP? So AMP is more. So what does it mean? What will happen to the concentration of ATP? If AMP is more, what happens to the concentration of ATP then? Yes? It is going to decrease, right? AMP means what? It is the, from ATP, the phosphorylation, the phosphate group has been removed, right? So ATP on hydrolysis will give you ADP plus PI, or it will give. So as I was saying, when you have got ATP, so ATP will give you on hydrolysis ADP plus PI, or it will give you AMP plus 2PI, right? So what is happening when the concentration of your AMP is increasing? Obviously, the concentration of ATP is decreasing, right? So, but
Yes,
pyruvate kinase is helping in the formation of ATP. So when there is an increased concentration of AMP, it means in the cytoplasm, the cell needs more of ATP. So it is going to activate your pyruvate kinase more. Now it is clear?
Yes, ma'am.
A high AMP concentration activates your pyruvate kinase. Okay. If you have got high concentration of ATP, it means what? The cell does not need more ATP. So glycolysis should stop. Then it is going to inhibit the pyruvate kinase activity. Okay. Acetyl CoA, which accumulates when ATP is in rich supply, also inhibits pyruvate kinase. Now, acetyl CoA, so we know that acetyl CoA will enter your Krebs cycle and electron transport chain and give you more amount of ATP. So again, I don't want more ATP because the cell has got sufficient ATP. Then again, it is going to inhibit the pyruvate kinase. Okay. So now, if suppose you're making a table, then you can write as PK means plus is your AMP, plus is your ADP, negative is your ATP, negative is your acetyl CoA. Now it is clear?
Yes, ma'am.
So this is important for the regulation because whenever question comes as MCQ, they ask like this only, like if you have a high concentration of ADP and AMP, what will happen to pyruvate kinase? Whether it will undergo glycolysis or it will undergo gluconeogenesis, such questions are asked. Okay. Now, F16 bisphosphate, that is fructose 1,6 bisphosphate, activates pyruvate kinase in the liver. The second example of feed-forward stimulation. Alanine is a bioproduct of pyruvate, is an allosteric inhibitor of pyruvate kinase. Again, negative is alanine. Okay. Phosphorylation of pyruvate kinase is regulated by blood glucose level just like PFK. So high glucagon level means low blood sugar. Insulin means high blood sugar. Okay. So when you have got
High blood sugar.
When you have got sugar more in the blood, then only your insulin is released. When you have got low blood sugar, then glucagon is released. Okay. So high glucagon causes phosphorylation, which in this case renders the enzyme activity. The regulation is at the substrate level. So these substrates are actually helping in the regulating the enzyme. Now let's discuss about the hormones level. Okay. So always remember insulin, it means it is high glucose level in the blood, right? So it means it should undergo glycolysis. Okay. It means it should activate your pyruvate kinase. This much is clear?
Yes, ma'am.
Okay. Next, if you have got glucagon, okay, so what glucagon is going to do? Glucagon means you have got low blood sugar level. So
Low blood sugar.
no glycolysis and gluconeogenesis. Gluconeogenesis means synthesis of glucose, right? So here the pyruvate kinase is inhibited. Okay.
Yes, ma'am.
Now, so this is the same thing which is shown in this figure also over here. So if you see low blood glucose level, it means what hormones will be active over here?
Glucagon.
Glucagon. And it is inactive. Your pyruvate kinase is going to be inactive then, right? Similarly, if you have got the okay, negative and all, this is about the pyruvate kinase here. So dephosphorylated pyruvate kinase means it is again active. If you have got the phosphorylated pyruvate kinase, then it is active. Okay. PK, if it is having phosphate group, it means it is inactive. PK is normal, then it is active. Itna clear hai?
Yes, ma'am.
Okay. Now hemolytic anemia. So pyruvate kinase deficiency also causes hemolytic anemia since RBC cells depend on the glycolysis for ATP synthesis. So if suppose pyruvate kinase deficiency is there, it causes accumulation of 1,3-bisphosphoglycerate and phosphoenol pyruvate. So the concentration of pyruvate and lactate are lower than the normal in this disorder. So the concentration is very less. Okay, so the ATP is also less. Acetyl Coa is allosteric effector for both glycolysis and gluconeogenesis. Acetyl Coa in high concentration inhibits pyruvate kinase and reciprocally activates pyruvate carboxylase. Now, where do you see pyruvate carboxylase? This we see in the case of your gluconeogenesis. Okay. So it is activated, pyruvate carboxylase is activated during gluconeogenesis. So acetyl CoA also inactivates pyruvate dehydrogenase, providing a regulatory link between the glycolysis and the citric acid cycle. Now, pyruvate dehydrogenase, what it does? After glycolysis, at the end, we get pyruvate, right? So pyruvates will give you acetyl CoA with the help of your pyruvate dehydrogenase. So in the next lecture, we'll discuss about the pyruvate dehydrogenase because it is like very vast. So when we say pyruvate dehydrogenase, it is again made up of around 3 to 4 subunits. Okay. So high concentrations of acetyl CoA are indicative of high energy supplies. Okay. Acetyl CoA ज़्यादा है, it means we have got high ATP source. So we don't want any more pyruvate kinase activity. So when the energy supply is high, metabolites are directed towards storage in the form of glycogen. Okay. Is this clear, Chandni?
Yes, ma'am.
Okay. So here actually the regulation of glycolysis gets completed.